So good morning students, welcome to medicinal chemistry classroom once again. So today we are going to study antiviral agents, which is a very important chapter. Antiviral agents are a class of medications used specifically for treating viral infections. Viruses are obligate cellular parasites composed of a nucleic acid core surrounded by a proteinaceous outer shell. Virus conducts no metabolic process on their own. They invade the host cell, which may be bacteria, animal, or plant cell. The virus turns the biochemical system of the host cell to its own purposes, completely subverting the infected cell. So, the structure of a virus. A virus does not possess cell wall. It consists of one or more of a linear or helical strands of either DNA or RNA enclosed in a shell of protein known as capsid. Capsid is composed of several subunits known as capsomers. In certain cases, the capsid may be surrounded by an outer protein or a lipoprotein envelope. Look at the schematic representation of a structure of a virus. So here you can see the capsid, capsomers, nucleic acids, and spikes, that is pepiomers. Moving on to the life cycle. So the viral life cycle, it varies with species, but they all share a general pattern which can be sequenced as follows. So the sequence of steps involves, first one is the absorption. So the adsorption, that is attachment of virus to the host cell. Then penetration, that is the penetration of virus into the cell. Then uncoating, then transcription, translation, assembly of the viral particle, and the release of the mature virus from the cells. Moving on to viral life cycle. So the viral life cycle, the first step is adsorption. What is adsorption? The attachment of virus to the host cell. Then penetration, penetration of virus into the cell. Next step is the uncoating step, that is genetic material or viral genome, that is DNA or RNA, passes into the host cell, leaving the capsid covering outside the host cell. Then comes transcription, the production of viral mRNA from the viral genome. Then translation, the viral genome enters the cytoplasm or nucleoplasma and direct or utilizes the host nucleic acid machinery for the synthesis of new viral protein and also for the production of more viral genome. The viral protein modifies the host cell and allows the viral genome to replicate by using host and viral enzyme. This is often the stage at which the cell is irreversibly modified and eventually killed. Next one is the assembly of the viral particle, that is the new viral core protein assembles into capsid and viral genomes. And the last step is the release of the mature virus from the cells by budding process or rupture of the cell and repeat of the process in fresh host cell. Since a host cell machinery is totally utilized for production of new virons, the normal cell function ceases at the time of replication. So this is just a schematic representation. So from this diagram you can understand the life cycle very easily. Then this is a list of some viruses and the diseases. So the picornavirus causes disease, polio and hepatitis A, rhinovirus, common cold and pneumonia, Toga virus, rubella and encephalitis, flavivirus, yellow fever, dengue fever and St. Louis encephalitis, Bunia virus, encephalitis, hemorrhagic fever, rhabdovirus, vesicular stomatitis, myxovirus, mumps and measles, Riovirus or rotavirus causes diarrhea, filovirus causes Ebola and Marburg, and retrovirus HIV. Then DNA viruses like herpes virus cause herpes and cold sores, Papua virus, polyoma and warts, adenoviruses respiratory complaints, then pox viruses cause smallpox and parvovirus, canine distemper. Antiviral agents are classified into three types. First one is classification based on chemical structures. Second one based on enzyme inhibition. Third one is based on treatment protocol. First, we will discuss about the classification based on chemical structure. So in this, first we have purine nucleosides and nucleotides. Examples: acyclovir, valacyclovir, ganciclovir, vidarabine, abacavir, famciclovir, penciclovir. Pyrimidine nucleoside and nucleotides: hydroxyuridine, trifluoridine, cidofovir. Next is thiosemicarbazole. Example is methizazone. Then adamantane amines. Examples: amantadine, remantadine, somantadine. Then we have interferons and miscellaneous agents like phoscarnet sodium and ribavirin. Second type of classification is based on enzyme inhibition. First one is inhibitor of DNA polymerase. Examples: hydroxyuridine, trifluoridine, vidarabine, acyclovir. Then reverse transcriptase inhibitors. Examples: zidovudine, salcitabine, didenosine, stavudine, lamivudine, abacavir. Then nucleoside antimetabolites. Examples: ribavirin. Then non-nucleoside reverse transcriptors. Example: nevirapine, efavirenz, delaviride. Then HIV protease inhibitors. Examples: saquinavir, ritonavir, indinavir, nelfinavir, ambrenavir, lopinavir. Third classification is based on treatment protocol. First one, treatment of respiratory virus infections. Examples: adamantane derivatives like amantadine, remantadine. Then treatment of herpes and cytomegalovirus infections. Purine nucleosides. Examples: acyclovir, ganciclovir, vidarabine. Then pyrimidine nucleosides, trifluoridine, hydroxyuridine, phosphorus derivatives, phoscarnet, sodium. Then treatment of HIV infections. Reverse transcriptase inhibitors like purine derivatives, didenosine, pyrimidine derivatives, zidovudine, stavudine, non-nucleosides, nevirapine, delaviridine, efavirenz, etc. Then protease inhibitors: saquinavir, indinavir, ritonavir, nelfinavir, ambrenavir, lopinavir. Then integrase inhibitors: sintenavir. So this is all about the classification. So it's very important. First we will discuss about the purine nucleosides and nucleotides. So first we have acyclovir. It's an inhibitor of DNA polymerase enzyme. Look at the trade names. The trade names are acyclovir, cyclovir, sovirax. Its IUPAC name is 9,2-hydroxy-ethoxy-methyl guanine. So the basic nucleus is guanine. What is guanine? Guanine is a nucleoside. It is composed of a pyrimidine ring and an imidazole nucleus. So pyrimidine plus imidazole. So these two fused ring structure is termed as guanine. So at the second position you have NH2, and sixth position you have keto group. And ninth position of this guanine is attached with 2-hydroxy-ethoxy-methyl group. So this is all about the structure of acyclovir. Moving on to synthesis. So starting from guanine. So guanine, upon treatment with chloromethoxy-methyl benzoate, so it loses one molecule of HCl to form an intermediate, which upon hydrolysis and reduction gives acyclovir. Moving on to the mechanism of action. Acyclovir is a synthetic analog of deoxyguanosine, in which the carbohydrate moiety is acyclic. The mode of action of acyclovir consists of the following consecutive mechanisms. And we have already studied the mechanism under product chapter, that is under site-specific bioactivation. So what happens is that acyclovir is first converted into its corresponding monophosphate form by the enzyme present in the virus, that is viral thymidine kinase. And this phosphorylation reaction occurs faster in infected cell than normal cell because acyclovir is a poor substrate for the normal cell thymidine kinase. Acyclovir monophosphate is further converted into di and triphosphate by the normal cellular enzyme guanosine monophosphate kinase. Then, acyclovir triphosphate inhibits viral DNA polymerase by competing with deoxyguanosine triphosphate. And the triphosphate drug is also incorporated into viral DNA polymerase, viral DNA, where it acts as a chain terminator. And this is a flow chart showing the mechanism of action. So acyclovir gets converted to active acyclovir monophosphate by the enzyme present in the virus, that is thymidine kinase. Then the active acyclovir monophosphate will be converted to acyclovir diphosphate by the enzyme guanosine monophosphate kinase. Then this diphosphate will be converted to triphosphate by the enzyme phosphate kinase. Then acyclovir triphosphate will it competitively blocks, or it acts as a competitive inhibitor or alternate substrate, or it acts as a chain terminator. And what happens is that the DNA chain gets terminated. And deoxyguanosine triphosphate is a nucleoside precursor used in cells for DNA synthesis. Its dose is 200 mg 5 times daily every 4 hours for 5 to 10 days, or 400 mg 5 times daily for 5 days in severely affected patients. And uses: it is effective against several DNA viruses including HSV1, HSV2, and against varicella zoster virus. An advantage is that uninfected human cells are unaffected by the drug. It is also used for short-term treatment of shingles and chickenpox virus, chickenpox caused by varicella zoster virus. Next is valacyclovir. Valacyclovir is a prodrug of acyclovir. It's a L-valyl ester of acyclovir. It is active against HSV1 and varicella zoster virus. Please find out the structural difference between acyclovir and valacyclovir. What is the difference? At the 9th position, right? So 9th position is attached with a little bit lengthier group. What is that? It is actually L-valine. L-valine. At the extreme end, it has the valine amino acid, and valine amino group is present, and rest is the same, right? So CH2 ethoxy group and methyl group is present. So L-valine 2 amino 1,6 dihydro 6 oxo purine methoxy ethyl ester. So this is the IUPAC name. No need to worry. Just write the structure and study. Then ADR, vertigo, confusion, dizziness, edema, and arthralgia. And its dose for the treatment of herpes zoster, the usual dose is 1 gram 3 times a day for 7 days. For the treatment of an initial episode of herpes, the usual dose is 1 gram twice daily for 10 days. And for the treatment of recurrent herpes, the usual dose is 500 mg twice daily for 5 days. And uses: used for the treatment of herpes zoster in immunocompromised patients. So dear students, these are the structures of nucleobases and nucleosides. So first of all, you by heart all these structures and the numbering also. So we will continue the second part in the next class. Thank you.